Cell theater, meiosis. Starring Peter and Effie.
Hey Dexter, what you doing?
Dee Dee, go away. I'm about to complete a fantastic discovery.
What is it?
Okay, I'll tell you. I've discovered that cells go through a process called meiosis.
Oh, that's when cells divide, reducing the number of chromosomes in the new cells by half. And then these new cells can combine and form new organisms later in life, obviously.
Well, the process is much more complex than that. These things called chromosomes are important and carry all the information about the cell. There are 22 pairs of identical chromosomes in each normal cell called homologous chromosomes. Smart people call a pair of such chromosomes synapses. And each pair is called a tetrad.
Well, did you know that there are three stages? No, did you know that there are two stages of cell division called meiosis 1 and meiosis 2, and that cells that are about to do meiosis have to go through the G1, S, and G2 stages of interphase first, which means that the cells start with duplicate sets of chromosomes. Because they divide twice, the final product will be 4 cells instead of just 2.
Well, in meiosis 1, there are different steps, such as prophase 1, metaphase 1, anaphase 1, and telophase 1, and cytokinesis 1.
In prophase 1, DNA wraps tightly together and forms these chromosomes. And these things, which I think probably look like spaghetti called spindle fibers, they show up. And then the nucleus and the nuclear membrane just disappear. The chromosomes go through synapsis, and the genes on each chromosome just match up with each other.
Yeah, but there's more. Chromatid detach part of themselves and give it to the other chromatid in the homologous pair. That's called crossing over, and results in genetic recombination because a new mixture of genetic material is created.
Okay, well, in the next step, metaphase, the tritards, what a funny word, line up in the middle of the cell like soldiers. And then the spindle fibers from each end of the cell attach to the center of each chromosome, and some from the other end attach to the others and pull each other away.
Ugh, fine. Then in anaphase 1, each homologous pair chromosome move to opposite sides of the cell. They do it randomly, and the separation is called independent assortment. This results in genetic variation.
The telophase numero uno, the chromosomes get to the opposite side of the cell, and cytokinesis starts. The new cell have a haploid number of chromosomes. There are 2 new cells, each containing half the number of chromosomes, but each having 2 copies as chromatids, because the first cell copied its DNA before meiosis 1.
What about meiosis 2, huh? It occurs after meiosis 1 and in prophase 2. Spindle fibers start to move the chromosomes to the midline of the cell, and metaphase 2. They finish and each chromatid is facing a different side. Then, in anaphase 2, they separate to go onto the different sides of the cell.
Okay, well in telophase 2, a nuclear membrane forms around each chromosome, and in each new cell, cytokinesis 2 happens, and then 4 new cells are made, but they have half the original cell's organelles.
Well, you should know that meiosis is the primary event in the formation of gametes, and the only things produced by meiosis are gametes in the reproductive organs.
Well, meiosis can form sperm cells in a thing called spermatogenesis or something like that, or egg cells through oogenesis, another funny word, which produces 1 mature egg cell and 3 just other useless things that just get ejected from the cells, but they're called polar bodies.
Fine. One last thing. Sexual reproduction is the production of offspring through meiosis in the union of a sperm and egg. They're genetically different than both parents because the genes are combined in new ways. Did you know that?
I know everything, obviously.
If Dee Dee knew all that stuff, then everyone knows it, so I guess I didn't discover it.